Life. How did it begin? Before we examine the origin of life, first we need to address what even is life. As we learned in our first module, life has 5 basic requirements: a need for energy, organization into membrane-bound cells, genetic information, the ability to replicate, and the ability to change over time. Cells are the building blocks of life, as they contain all the necessary organelles that perform all these functions. But cells cannot survive on their own. What keeps them functioning, you may ask? Proteins. They maintain cell structure, perform transport functions, and carry out many of the enzymatic reactions in modern cells. Proteins are a large portion of what keeps our cells functioning. But what are they made out of? Amino acids. Amino acids are the building blocks of proteins. It's like a puzzle inside a puzzle inside a puzzle. On a basic level, an amino acid has 4 parts: an alpha carbon, the hydroxyl group, and the amine group, and the side chain. The 20 different side chains are the difference between the 20 amino acids. Amino acids link together to form a peptide backbone, and once large enough, become what we call a protein. In 1953, Stanley Miller proved that amino acids could form abiotically under the conditions of the primordial soup, an atmosphere of methane, ammonia, water, and hydrogen, as opposed to one of carbon dioxide, nitrogen, oxygen, and water we have today. Soon after this, Fox and Hardis showed that these amino acids, when condensed under high heat, can form peptides, which can then polymerize into proteins. Given the pre-life conditions, this proves that simple organic molecules can polymerize to become complex macromolecules. Proteins have many different jobs in a cell and are essential for a wide variety of cell functions. For life to have evolved, there needed to be a wide variety of polypeptide chains with useful functions, because the chance of a random polypeptide sequence being useful is very low. Researchers at Georgia Tech sought to prove that there is a large sequence of diversity that could be and are formed out of only a few amino acids through the use of disopeptides, a type of peptide backbone that contains ester and amide linkages in the peptide backbone. The disopeptides are capable of chemical evolution due to their ability to change the linkages in their backbones. They studied disopeptides, diversity, and how to change in dry environments. They used 3 amino acids and tested how 3 different amino acids affected the diversity of the amino acid sequences. They did this by using mass spectrometry and multidimensional separations to categorize the amino acids into different sequence mixtures of protopeptides. They found that abiotic processes can and do generate a huge diversity of polypeptide chains, making it more plausible that some of them may have functions useful for cellular life. This experiment showed for the first time that a huge diversity of peptides can be formed from a few ingredients, thus proving it is possible to create a large enough diversity of peptides to make it extremely likely that the perfect combination of proteins necessary to form life can be made.